Welcome biologists. Today we're going to be looking at mechanisms that control gene expression. This is taken from the cellular control topic on the OCR specification for A-level biology. And as you can see here, there are three main ways in which we can control gene expression. So we're going to start off with the transcriptional level, and this is looking at the Lac Operon. Now, the Lac Operon is normally used in bacteria, and this is used to regulate the expression of enzymes that break down lactose. Now, normally a bacteria would undergo and use glucose in aerobic respiration as a respiratory substrate. However, when glucose isn't present and lactose is present, it would prefer to use, obviously, lactose because there's no glucose present. So in order to break down that lactose, the bacteria needs to produce enzymes to hydrolyze that sugar, and it also needs to produce proteins to transport the lactose into the actual bacteria itself. Now, there's no point in the bacteria making these enzymes and proteins when lactose isn't present because it's just wasting resources. So it's got this very, very clever Lac Operon, which determines if I'm going to be making these enzymes or not. So first of all, I have this regulatory gene, and the products of this regulatory gene switch on or off the other gene. So in this particular case, my regulatory gene codes for the repressor protein. We also have the structural genes, which are these ones here, these red boxes here that follow on. And these red boxes, the products of these red boxes are the enzymes, which in this case is beta-galactosidase. And this is one of the enzymes that helps to hydrolyze or break down the lactose. We also have the operator region, which in this particular case is where the repressor protein binds to. And then we have the promoter region. And the promoter region, this is where RNA polymerase ideally would like to bind to. So how this works is that when lactose is present, lactose binds to this repressor protein. Now, as you can see in this top image here, the repressor protein is normally bound to the operator region, blocking, therefore, the promoter region. Therefore, RNA polymerase cannot access this promoter region. However, when lactose is present, it binds to the repressor protein. The repressor protein changes its shape and therefore no longer binds to the operator region. So it moves away from the operator region. This then therefore allows RNA polymerase to bind to my promoter region and transcribe these structural genes to make my enzymes. Now, this is really important because these enzymes are then needed to use to hydrolyze lactose and also transport it into the cells. Transcriptional factors also occur in eukaryotes, and you'll meet a couple of these later on, but CAMP is a very, very, very important. Cyclic AMP is very, very important here in acting as a transcriptional factor. But there are many, many, many things that act as transcription factors in eukaryotes. You just need to be aware that in eukaryotes, genes can be turned on and off. So we're now moving on to the next part, which is the post-transcriptional level. So after post-transcription, I've obviously got my mRNA. Now, we need to be aware here that DNA contains regions of coding and non-coding DNA. So my non-coding regions are called introns, and my coding regions are called exons. So when I make my mRNA from my DNA, I'm going to have regions of my mRNA that contain this non-coding DNA. And in order to get rid of them, I have to go through splicing. Splicing is the name of this process. And this again is activated by CAMP or cyclic AMP, which makes a protein, which triggers a protein to be made to cause this splicing. Now, splicing is where mRNA is modified by the removal of introns to produce mature mRNA. Now, it's worth remembering here that my mature mRNA, this image at the bottom here, this could change depending upon if the splicing process changes and different versions of that mRNA could actually be produced. So that's our post-transcriptional level. The last one is our post-translational level. So at this point, I already have my proteins. And my proteins can be modified by the triggering of hormones or sugars, which binds the cell surface membrane and trigger this cyclic AMP to become produced. And cyclic AMP can alter the structure of my proteins. It might do this by activating something called protein kinases, which then can add phosphate groups or phosphorylate my protein. Another way in which cyclic AMP might work is to activate enzymes, so turning a pre-active precursor into an active enzyme. So there we have it, the regulatory mechanisms that control gene expression. Good luck with your exams.